Greetings everybody, I am Shujat Ali from Medicos Lectures by Shujat, and today we are going to talk about GIT histology. Means we are going to study gastrointestinal tract at a tissue level. So whenever we are going to study something at a tissue level, we have to know its layers and also the layers sublayers. So in case of GIT we have four major layers: mucosa, submucosa, muscularis externa and serosa or adventitia. Mucosa is the outermost layer. It contains further three major layers, which are epithelium, lamina propria and muscularis mucosa. In the same way, submucosa contains blood vessels, lymphatics and nerves. And muscularis externa is further divided into two layers, outer circular and inner longitudinal layer. And serosa or adventitia, it contains mesothelial cells and is retroperitoneal. Now talking about this very first layer of mucosa, the epithelium and its role in GI tract. Epithelium. Epithelium is stratified squamous in esophagus, stratified columnar without goblet cells in stomach. In small and large intestine it is stratified columnar but with goblet cells. And in case of anal canal, there is a slight change in that, means it varies from surfaces. We have stratified columnar, cuboidal, non-keratinized and keratinized. Talking about the roles of epithelium in our GI system. So first role is protective role, means it protects GI from pathogens and antigens. Second role is absorptive role. We have microvilli, villi, which are mucosal projections, and we have lica circularis, which are submucosal projections. We have glycocalyx for absorption and disaccharide digestions. So they basically play a role in absorption. Lica circularis, glycocalyx, villi and microvilli, they absorb the nutrients from that. In the same way, there is some secretory role if there is an absorptive role. So secretion are basically by mucus, by enzymes, by antibodies. They are going and perform their secretions. We also have glands. We name them as Brunner's gland, which are playing role in alkaline secretion, and extramural gland, which are basically present extracellularly. So we name them that extramural gland. Now talking about further layers. So in muscularis mucosa, it is basically a thin layer of smooth muscles having inner circular and outer longitudinal layer. Our main point is lamina propria. Lamina propria is basically next to that of epithelium. Epithelium is outermost layer, then lamina propria present, then muscularis mucosa occurs. So lamina propria basically it contains some glands, some cells from esophagus to small and large intestines. So in esophagus it contains cardiac glands, in stomach it contains gastric glands, which are possessing chief cells having main role in secretion of HCl and pepsinogen. In the same way, in small intestine there is different cells having different roles, like we have Penneth cell, we have crypts of Leberkuhn, muscularis externa as well as lamina propria. When we basically see the structure, so muscularis externa, then we have lamina propria. On that lamina propria we have muscularis, we have Penneth cells and we have crypts of Leberkuhn. Talking about the Penneth cells, we also have pair patches and M cells. Talking about Penneth cells, they are basically special type of cells which secrete lysosomes and alpha defensin. And pair patches, they are large lymphoid nodules. This basic structure, we have a lymphoid nodule, and when they become a very large, we says that we have a pair patch. On surface of these lymphoid nodules, these pair patches, we have M cells that are going to be present. We also name them as microfold cells. So what's the difference between small and large intestines? Large intestine didn't have intestinal villi, while small intestine possessing intestinal villi. Crypts of Leberkuhn is present in small intestine as well as in large intestine, while Pennant cells, which have main role in lysozyme secretion and alpha defensin, is absent in large intestine, while in the same way Pennant cell is present in small intestine. About further details, so we have submucosa, which is possessing large blood vessels, nerve plexus, and occasional glands. Occasional means which are present, which are not present all around, all throughout the organs. They are present occasionally at a distant locations. Nerve plexes are basically are of two types: submucosal or mesenteric, and mesenteric or Orbeck. Talking about submucosal or mesenteric, so it contains pre and post parasympathetic nerve fibers, as well as post sympathetic cell bodies, as well as post ganglionic fibers. And mesenteric or Orbeck, it contains pre ganglionic parasympathetic cell fibers, post ganglionic. In the same way, it contains a supply that is mainly concerned with vagus nerves. Muscularis externa, it is divided into two layers. In initial we discussed it. Inner is circular, outer is longitudinal. Inner layer has a role in mixing movement, and outer has its role in peristalsis. And Haustra is basically a ribbon like arrangement of large intestine like this. So we says that it is Haustra. Talking about the last layer, which is basically the outermost, serosa and adventitia. Serosa, it contains mesothelial cells plus connective tissue, and adventitia, it is made up of only connective tissue, no mesothelial cells. Outermost part of GIT, and it is attached with abdominal cavity, pelvic cavity, and with connective tissue of adventitia. So guys, this is our basic overview lecture, overview of layers and sub layers of organs and complete GIT system. Hope you all will understand it. Don't forget subscribe Medico's lectures by Shujat. Thank you so much.